This question is on monoclonal antibodies. So a person with late-stage HIV infection has AIDS, and scientists have produced monoclonal antibodies for HIV, and these can prevent a person infected with HIV developing AIDS. So you want to describe how the monoclonal antibody for HIV can be produced. So the way that would work is very similar to how monoclonal antibodies would be produced in general, but we want to make sure we're being specific in our answer. So first of all, we have the HIV antigen is injected into a mouse. You can use other mammals for this step as well. Then we are going to be extracting the lymphocytes, which make a specific antibody. And this will be complementary to HIV antigen. These are then combined so that you have your lymphocytes combined with tumor cells, and that forms a hybridoma. So you have hybrid in there because it's made of two different types of cells. And these are then, so your hybridomas are cloned. That means you have many cells which can produce the antibody. And for the next part of the question, we have figure 9, which shows how HIV enters a human cell. And we have the diagram here, and it says, suggest how the monoclonal antibody for HIV helps to prevent a person infected with HIV developing AIDS. So helps to prevent it. So for this question, we know that the monoclonal antibody that is present, the monoclonal antibody is complementary to the HIV antigen, just like what we wrote in the last question. And these attach to the HIV antigen. And what then happens is that this means the HIV antigen cannot bind to human cells / enter the human cells. So essentially it would stop this process from occurring. So if that's being stopped, it can't enter the cell and the person won't go on to develop AIDS.